Okay, so today we're going to go through the material from Lab 9. This was the histology of the alimentary canal organ, and then we'll also have the histology of the liver lobule as our last model. Okay, so this is a model of the histological layers of the small intestine. It's specifically the duodenum. And we can tell that if we kind of swing the model up this way, because you can see that there's a big gland here in the submucosa, and that is one of the duodenal glands. So we know specifically that this is a model of the duodenum. We would have been able to tell it was a small intestine regardless, because you can see that you have nice villi here, those nice finger-like projections. Okay, so we're going to start from the luminal side and work our way down to the peritoneal side. So the layer that's closest to the lumen is the mucosa, and the mucosa has three components: the epithelium, which you can see nicely here, the simple columnar epithelium, and these little blue structures that you see here are goblet cells. Okay, and then the gray material that you see within each of the villi and in this space between the villi and this next pink layer, that is the lamina propria. And the lamina propria is the special name for the areolar connective tissue layer in a mucous membrane that's associated with an epithelium. Okay, and last but not least, in the mucosa, we have the muscularis mucosa, that little layer of smooth muscle, fairly thin, but it helps the mucosa to move, to dislodge particles that might be stuck between the villi, for example. Okay, in the villi, you can also see that we have blood capillary beds associated with absorption. Here's the red and the blue of the arterial side of the capillary bed and the venule side of the capillary bed. And then the green here would be the lacteal that would be helping us with lipid absorption. You can also see that there's a lovely lymphoid nodule in here that would be part of the MALT tissue, that mucosa-associated lymphoid tissue. Okay, so here was the border of the mucosa. As we move past that, we get to the submucosa. It's again gray, showing us that we have areolar connective tissue here. You can see lots of blood vessels here. And again, we have that submucosal gland, the duodenal gland. So this is the duodenum that we're looking at. Okay, and here's a lymphatic structure, and then you can see the yellow of the nerve plexus as well. As we move past that, we get to the muscularis externa, and in the small intestine that has two layers. Okay, the circular layer, which is closer to the lumen, and the longitudinal layer, which is closer to the peritoneum. Okay, both of these composed of smooth muscle. Okay, and then we get to our final layer, which is the layer that is actually facing the peritoneal cavity. This is the serosa layer, and the serosa is also the visceral peritoneum, so it's going to have that serous membrane structure of the simple squamous epithelium and the thin layer of areolar connective tissue next to it. Okay. Okay, now we have a lot of the same, really all of the same layers on this next model, but this model is organized so that it shows you how those layers are modified as we move from one organ to the next. Okay, so this top section is representing the esophagus. This next section is representing the stomach. The third section, the small intestine, and then the bottom section here is the large intestine. Okay, so I'll kind of turn it sideways here for a moment so we can look at the layers. Okay, so again, we're going to have mucosa, okay, with the epithelium, lamina propria, muscularis mucosa, submucosa, muscularis externa, and then we would have a serosa here, but since this is the esophagus, instead of a serosa we have an adventitia here. Okay, some other modifications that you can see specifically associated with the esophagus are the esophageal gland here in the submucosa, releasing that lubricating material that helps the bolus to move through. And then we also have our non-keratinized stratified squamous epithelium that helps us with the friction, abrasion, and heat protection that we need here in the esophagus. Okay, moving to the stomach. Okay, now, again, epithelium, lamina propria, muscularis mucosa. We've switched to simple columnar epithelium. That's mostly goblet cells or mucus cells. Submucosa again. It's not as obvious here, but in the stomach we should have 3 layers of muscle, smooth muscle, okay, to help us with that mechanical digestion. And now we have our serosa. Okay, small intestine, lots of villi obvious, simple columnar epithelium, lamina propria, muscularis mucosa, all the parts of the mucosa layer. Submucosa, 2 layers of smooth muscle in our muscularis externa, and a serosa. Okay, and last but not least, the large intestine. And the large intestine, again, we have simple columnar epithelium, a lot of goblet cells here to make the mucus, to lubricate the food residue as it's moving through the lumen. So epithelium, lamina propria, muscularis mucosa again, all the sublayers of the mucosa, submucosa, and then I'm going to switch over to this side actually because you can see it better over here. Once you get to the large intestine, you have a complete circular layer but an incomplete longitudinal layer. So this darkened area here, and I'm going to kind of flip it on its side so you can see it a little better. See, that darkened area there is representing one of those little strips of longitudinal muscle, the taenia coli, that are all that's left of the longitudinal layer of smooth muscle. Okay, and then we're still, at least in some sections of the large intestine, intraperitoneal, so we have our serosa here too. Okay. All right, so those are the layers of the alimentary canal wall. Now we're going to move to the histology of the liver, and each of these hexagonal structures, you can see one here very nicely, is a liver lobule. At the corners of the liver lobule we have a portal triad, and each portal triad is going to contain 3 vessels, a branch of the hepatic portal vein, a branch of the hepatic artery, and the green is a bile duct. Okay. And any blood that is entering the liver lobule is going to be coming from these 2 blood vessels in the triad and heading through the lobule in this direction toward this vein here, which is the central vein, appropriately named. Okay, so at this point we're going to switch over to this section, which is, if you could imagine, you took a wedge out of this lobule and then enlarged it here. Okay, so here's the portal triad on this side. Again, the bile duct in green, the branch of the hepatic portal vein in purple, the branch of the hepatic artery in red. And here's our central vein over here. Okay, so blood is coming from these 2 vessels, the branch of the hepatic artery is oxygen-rich blood. The branch of the hepatic portal vein is oxygen-poor but nutrient-rich blood. This blood is going to flow together into these capillaries, which are running through the lobule, and these are the liver sinusoids. Okay, now as the blood is flowing through, two kinds of cells have access to the blood. Most of the cells that are forming the structure of the liver are these tan cells, and those are the hepatocytes. And they're the ones that are going to process the nutrients in the blood, remove any lipid-soluble vitamins, detoxify chemicals that are coming through, and they're also going to make bile, which we'll talk more about in a moment. Now, we're also going to clean the blood as it's moving through and remove any particulate matter. That's going to be the job of these pinkish cells that you see inside the sinusoids. These are the hepatic macrophages. Okay, and like any other macrophage, they're experts at grabbing solid material and pulling it out of their environment. So these are going to be our big cleanup cells. Okay? Now, we said we're also going to be making bile in the hepatocytes. This green structure that you see here that looks kind of like a set of railroad tracks on a map, that is a bile canaliculus. Okay, so all of these hepatocytes that are here are making bile and putting it into the bile canaliculus, and that bile is eventually going to drain into the bile duct. Okay, so blood moving through the lobule enters at the portal triad and leaves through the central vein. Bile is being produced in the lobule and drains in the opposite direction toward the bile duct.